Today, we will learn molecular docking interaction analysis using AutoDock, UCSF Chimera, and LigPlot. Open AutoDock Tools and click on Analyze, click on Dockings, then Open. Select the generated DLG file from your working folder. After loading, the DLG file will open and display the number of docking conformations generated during the docking simulation. Again, click on Analyze, click on Macromolecule and then click Open. Load the target protein PDB-QT file for visualization of the protein-ligand docking interactions. For better visualization and interpretation of docking interactions, display the macromolecule in ribbon representation in AutoDock Tools. Now, click on Analyze, click on Conformations and then click on Play, ranked by energy to visualize the docking conformations arranged according to their binding energy values. A small window will appear. Click on Open Panel to access the play options. A new Set Play Options window will open. Check the options Show Info and Build Hydrogen Bonds for better visualization and interaction analysis. You can drag and arrange the opened windows on the screen according to your convenience for better visualization and analysis. The opened hydrogen bond window will display the hydrogen bonds present in the selected docking conformation. The conformation info window will show various parameters, including binding energy, key value, intermolecular energy, and ligand efficiency. You can record these values in your result table for further analysis and comparison. Using the Play Conformations window, you can move forward or backward to observe different docking conformations. You can also click on Play to visualize all predicted ligand binding conformations sequentially. For example, if you want to save the first conformation with the lowest binding energy, click on Build Current and then click on Write Complex. A new window will appear where you can save the protein-ligand complex with a suitable file name. Add the .pdb extension before saving the file. This will save the selected docking conformation in PDB format for further visualization and interaction analysis. Now open LigPlot to visualize the 2D protein-ligand interactions. To load the saved conformation PDB file in LigPlot, click on File, click on Open, then click on PDB, MMCIF file and select the saved protein-ligand complex PDB file. A separate window will appear displaying the detected ligands present in the complex structure. Select your ligand molecule and click on Run to generate the 2D protein-ligand interaction diagram. In the LigPlot diagram, hydrogen bonds formed between the ligand and amino acid residues are represented by dotted green lines, while hydrophobic interactions are shown by semicircular or semilunar arcs around the ligand atoms. Here, you can manually arrange the amino acid residues involved in hydrogen bonding and hydrophobic interactions according to your visualization and presentation requirements. You can also turn the chain labeling on or off and adjust the font size of the amino acid residues for better visualization and figure presentation. If you want to compare your control docking conformation with the docked ligand molecule, click on the Add button to load the ligand conformation PDB file. After loading the PDB file, use the split-screen view to visualize both interaction diagrams simultaneously and analyze the similarity in binding interactions, hydrogen bonds, and hydrophobic contacts between the control ligand and the docked molecule. Similar interacting amino acid residues present in both conformations will be highlighted and encircled in red for easy comparison and interpretation of binding similarities. After adjusting the interaction diagram according to your requirements, you can save it as a PS, PostScript, file or capture a screenshot. For better image quality and higher resolution, save the diagram in PS format and then convert it into JPG or PNG format for publication or presentation purposes. Now we will analyze the saved protein-ligand docking conformation using UCSF Chimera. After loading the complex structure in Chimera, you can generate high-quality visualization images of the docking complex. Click on Select right-pointing arrow residue and select the ligand molecule. Then, use the Presets option to visualize the protein-ligand complex in different colored representations for better structural interpretation and presentation. To change the background color to white, click on Actions, click on Color and then click on All Options, click on Background Color, and select white. You can change the appearance of the ligand by going to Actions, click on Atoms, then click on Ball and Stick to display the ligand in ball and stick representation. Now we can visualize the hydrogen bonds formed between the ligand and the target protein. Go to Tools, click on Structure Analysis and click on Find Hydrogen Bond. A separate window will appear. In this window, check the options, Find Only Intermodal Hydrogen Bonds, and Label hydrogen bonds with distance to display hydrogen bond interactions along with their bond distances. To save the current visualization image in high resolution, click on File, then click on Save Image. A separate window will appear. Check the Use Print Units option to enable print resolution settings, enter 300 dpi as the resolution, and save the image in your desired folder. Now we will also analyze the DLG file to interpret the molecular docking results and identify the best docking conformation based on binding energy and clustering analysis. Open the generated DLG docking log file using Notepad or any text editor for manual analysis of docking results. Scroll down through the file to locate the sections titled Clustering Histogram and Lowest Energy Docked Conformation from each cluster. The Clustering Histogram section provides information about the grouping of docking conformations based on RMSD values and helps identify the most populated and stable binding conformations generated during the docking simulation. In the section Lowest Energy Docked Conformation from each cluster, you can observe important docking parameters such as binding energy, kilocalorie per mole, estimated key value, intermolecular energy van der Waals and hydrogen bond energy, electrostatic energy, torsional energy. Generally, the conformation with the lowest binding energy and favorable interaction profile is selected as the best docking pose for further analysis. Record the binding energy, key value, cluster rank, and other important parameters in your result table for comparison and interpretation of molecular docking results.